Hi everyone. So we are going to go over some gene expression examples today. So this is going to help us practice using our terminology from what we learned about DNA. And we are going to learn a little bit about how genes can be controlled so that we're not always producing all of the proteins, because that would be crazy. So let's get started. So gene expression examples. So what is gene expression? So I want you to take a moment, do a brief check-in on iClicker, and let me know what you think gene expression is. So pause. Okay, so gene expression is how we control what genes are on or off. Okay, so think of it like a light switch. So, okay. Okay, so proteins, when we're talking about whether genes are turned on or off, we have these things called transcription factors. Transcription factors, if we look over here, are going to be proteins like this one and this one. They are proteins, and they help us decide if RNA polymerase is going to come in and bind and make our mRNA. Remember, mRNA is the messenger RNA that goes from the nucleus to the cytoplasm. Okay? So we're going to talk about this, an example in bacteria. So we know that bacteria can use sugar as fuel, right? We talked about the different types of anaerobic respiration that bacteria can use, and all of those used our sugar molecule glucose. Okay, so now we're going to talk about something that's a little bit different. And so this is, let's see. So this is what we usually use, glucose. And we typically have glucose. But there is another sugar that we sometimes see bacteria use, and that is lactose. Remember, the ending OSE means sugar. Glucose, lactose, they are both sugars. Okay? And so if we are using glucose and we don't have any lactose, we can keep this gene off. This is the gene called the lac operon, and this makes a protein to use lactose. Right, so we got lac, right? Okay, so we have our bacterial cell. We have our DNA here, and we have a promoter. Remember that promoter is where our RNA polymerase is going to bind to start transcribing the gene. Okay, so typically we have these things called repressor proteins. And if the repressor protein is here, if it is stuck onto this gene, then we cannot transcribe, right? The repressor is like a roadblock here. Repressor is a roadblock. Okay, so if we can't transcribe, we don't get any mRNA. And if we don't have any mRNA, we don't get a protein. Okay, so if there is no glucose, which is our main fuel, but we have lactose. This is a situation where that gene is going to be turned on, okay? So lactose being present, we can see it right here. It is going to bind to the repressor protein. So we have it binding right here. And when lactose binds to the repressor protein, it can't bind to the DNA, right? So now our RNA polymerase has the ability to bind to that promoter and begin transcription. Once we can transcribe, we can get our mRNA product, which then leaves the nucleus, gets translated, and then we get our protein product. Okay, and that protein product is an enzyme called lactase. Lactase. Remember the ending ASE equals enzyme. Okay, so lactose is the sugar, lactase is the enzyme, and lactase is what breaks down lactose. So, two very similar words, but they have different jobs. Okay? So, how does a bacteria whose ability to break down lactose can be turned on and off, how does this relate to human lactose intolerance? So go ahead and take a moment to think about this in iClicker. You can pause the video, type out your answer, and then we're going to move on. Okay, so let's see if this video will load real quick. Humans, when they are young, are able to digest lactose, like most mammals. Okay. So if we go here, let's see. So humans under age 5 can make their own lactase, right? So humans have a gene for lactase, and it has this promoter. And if this promoter is active, then we can make the lactase mRNA and then make the lactase enzyme. So go ahead and answer this iClicker question. What is lactase? And this, for this one, just select any correct answer. Okay, so lactase, remember, is an enzyme, and an enzyme is a type of protein, okay? Okay, so now let's talk about this promoter. What is the lactase promoter? Is it a protein, a DNA sequence, or an mRNA? And we're talking about this right here. Okay. So this promoter is a part of the gene, right? So this sits here, right? And this is DNA. And the promoter is where our mRNA can bind. So we'll do this. We've got our mRNA, not our mRNA, excuse me, our RNA polymerase, which is also a protein. Okay, our RNA polymerase can bind. It can then transcribe our gene. Remember, when we are transcribing, we are reading the gene and making an RNA copy. Well, complement, say complement. Okay. So we have our lactase mRNA that gets taken out of the nucleus and made into an enzyme. Okay. Now, there is a mutation in humans, like the video is saying, where this lactase enzyme is produced after age 5. At about age 5, lactase typically is no longer produced in humans because humans would probably not need to digest milk after infancy, right? Okay, so in we'll say, let's see, in majority humans, we do not see lactase as an adult. Okay. However, sometimes we have a mutation. And what is a mutation? A mutation is an altered version of a gene. So, in this mutation, these humans have a version of the lactase promoter that continues to work. So, most of the time, so this would be in babies, right? We get the production of lactase mRNA, but in adults, we are not going to have an active promoter, so we don't get any mRNA. In the mutant version of this gene, we have a mutant lactase promoter. This can continue in adults. So adults producing lactase. Okay? So let's think about this for a moment. We have majority humans who do not produce lactase and cannot digest milk. Then we have mutant humans who can digest lactase because they have a mutant promoter that allows them to produce their own enzyme lactase. Okay, so remember, originally we talked about the lactase gene in bacteria. So if we have bacteria growing in glucose, are we going to see transcription and translation of lactase? So the answer to this is no. Remember, they keep the genes off. Right? Now, if we have lactase that is present, lactose, the sugar, we have lactose, the sugar, present, then we will see lactase transcription and translation. In human infants, remember, human infants need to be able to digest lactose, so they will be transcribing and translating lactase. Okay. Now, human adults with a regular lactase promoter. Now, take a moment and answer this one in iClicker. Okay. All right. So, we know that humans with the mutant lactase promoter can digest lactose. They have the enzyme lactase. So their transcription is yes. So think again, do our humans with the regular lactase promoter transcribe and translate lactase, the enzyme? Okay, next we have bacteria in the human gut. So if a bacterium living in a human gut is just hanging out, and then their human drinks a glass of milk. If that human has a regular lactase promoter, is this bacterium going to be transcribing and translating the lactase gene? Okay, Our human has a regular lactase promoter. So we go back up here. Are they making lactase? No. Ooh, let's go back. So there is lactose present. Oh my goodness, I keep pressing the wrong button. There's lactose present. So if there's lactose present, remember no, that's not going to show up very well. Our bacteria is going to turn that gene on. Okay, now bacteria living in a human adult gut who have a mutant lactase, remember the mutant lactase promoter allows lactase expression, so that human is not going to have any lactose left in their gut. Okay, and remember, if there's no lactose, there's no bacterial expression, right? So no. Okay, now this next one, let's go back here. Sorry. This next one, go ahead and answer these 3 in iClicker. It's going to be a short answer, so just do a 1, a 2, and a 3. Are these 3 humans going to be considered lactose intolerant? Lactose intolerant. Okay, go ahead and answer that in iClicker. And now we will discuss. Where is my pen? There it is. Okay, so human infants. Are human infants considered lactose intolerant? Remember, that means not. So they are not lactose intolerant, means lactose not tolerant. So babies have lactase, so they are not lactose intolerant. They can tolerate lactose. Humans with regular lactase promoters are not making their own lactase. They would be lactose intolerant, right? So they leave their lactose lying around, and then those bacteria start fermenting the lactose sugar. They start breaking down that sugar. Okay. And then lastly, our human adults with a mutant lactase promoter, they have the lactase enzyme, so they would not be lactose intolerant because they can break down their own lactose. Okay. So how does this produce the symptoms that we see in lactose intolerance? So what does this mean? So if N means not tolerant, let's say we have this guy right here has our mutant promoter. So he's making lactose, right? We have he's making lactase. Oh my gosh, excuse me. He's making the enzyme lactase. He can break down lactose, the sugar. If all of that lactose is broken down, the bacteria keep their genes turned off, right? The repressor is bound to the gene because there's no lactose, right? There's no lactose present. Okay. Now, on the other hand, if we have someone with the regular lactase promoter, that means that that human is not going to be producing the lactase enzyme. And this means that lactose is present in the gut. Lactose in the gut. Okay. Now that lactose, this diagram is in bacteria. By the way, these are the gut bacteria. Gut bacteria. Okay. Now that lactose binds to the bacterial repressor protein and makes that repressor unable to block RNA polymerase. Now, RNA polymerase can transcribe mRNA, and it can transcribe the lactase protein. So now our bacteria have lactase. And when bacteria have lactase, they can start to ferment sugars. And what happens when we have fermentation? We have these genes turn on to break down the lactose, and then because those bacteria are fermenting sugars, we have the production of byproducts like gas, and that gas can cause things like the cramping and stomach pain that some adults experience when they ingest lactose. Okay. So let's talk about one more example. So this example is going to be about cats. Okay. So there is a type of cat. This is called a Sphynx cat. And Sphynx cats are hairless. You can see a little bit of hair here, but they are considered a hairless cat, right? And I personally, I think these cats are cute. But some people think that they are not cute. Just as a little fun eye clicker, go ahead and tell me if you think that Sphynx cats are cute or not cute. So Sphynx cats they are the product of a mutation in the hair growth genes of cats, right? So they're a little bit, their history is a little bit muddy. So there are sort of two sources. There was a breeding program where a hairless cat was born, and then that breeder worked on stabilizing that gene so that it could be reliably reproduced so that they could have hairless cats as pets. That was not super successful. And actually, there was a second mutation that was found in a different set of cats. This mutation was a bit more stable. And so between the breeding program and the second population with different mutation, we have seen a more stable population of hairless cats, and we are able to keep them as pets now. So there are some really good videos about the history of this hairless cat mutation, and there's a lot more information about it on this website right here if you want to read about it. But let's talk about how this expression changes. So we were talking before about repressors that keep the mRNA from being transcribed. But there can also be mutations that change the way the mRNA is processed. Remember, we talked about processing or splicing. So take a moment and answer this question. What is splicing? Is it A, removing exons from RNA, B, removing introns from RNA, C, removing amino acids, or D, removing DNA? Take a moment, give your answer, and we can discuss. So remember, exons are expressed. Okay, exons are expressed. Introns are in the way. Okay, so if introns are in the way, then that is the part of the RNA that we want to remove. Okay? Okay, so here's our little intron. Boop, boop. Now we have this section, or this GT. So at a normal splicing site, we get a GT. At an altered splicing site, we get an AT, right? So that's a change in one nucleotide, but it makes a big difference. So in this normal keratin mRNA, we get the intron 4 taken out, right? So intron 4 gets removed, and we see exon 4 and exon 5, okay? And there's nothing in between them. In our altered keratin mRNA, this intron stays. Okay, so we can see this intron over here. Now we have exon 4, intron 4, and exon 5. This makes our mRNA a little bit longer, right? However, this intron 4 contains a stop codon. This is a sequence of mRNA, 3 nucleotides, that codes for the end of a particular process. And go ahead and answer this iClicker question. Which process stops when we see a stop codon? Remember what we had in our last set of slides, okay? Codons are part of translation. Okay, codons are part of translation. So if we have a stop codon, we are stopping translation. Okay. Now, we said the mRNA of the altered keratin is longer. However, here we have this stop codon in intron 4, right? In our normal keratin, our stop codon is here. This is our stop codon. Okay? So if our stop codon is at the end here, then we have amino acids being added all the way to the stop codon. In our altered mRNA, we still have this stop codon, but we also have this stop codon. So in our altered mRNA, our ribosome is going to read, it's going to get to this stop codon, and it doesn't just skip a stop codon. That stops the process entirely. So our protein from the altered keratin is shorter than our protein from the normal keratin. So the protein is shorter in our Sphinx cat because our Sphinx cat has the altered keratin mRNA. Okay, so with this protein being so short, the keratin is weaker. So the hair in the Sphinx cat is weaker. So it's typically shorter, it's softer, it falls out more easily. So this is why hairless cats, they may have these teeny tiny little hairs, but they don't have a full coat of fur like you see with cats who have normal keratin expression. Okay, so that is it for gene expression. Talk to you next time.